Don't ask me.
Okay, getting started. Yeah, I'm started recording. So the first thing is signal transduction for insulin or glucagon and epinephrine insulin. Regulation via covalent modification, allosteric effectors, and genetic regulation of enzymes are always factors in control of the rate or enzyme catalyzed reactions and their metabolic processes. Okay, how do we feel about that? Just for starters. Good, good. So do we need to go over any of it?
Maybe like a quick summary.
Okay.
Please through it.
So we have RTKs, receptor tyrosine kinases, and the G-coupled protein receptors, right?
Mm-hmm.
Cool. So the RTKs, they phosphorylize directly, right? They get activated, they phosphorylize, that phosphorylation goes and does whatever it needs to do. For the G-coupled protein receptors, it's an alpha subunit that gets taken off, and that goes out, and most of the time it's adenylate cyclase, which cyclizes AMP to make cyclic AMP, and in turn that makes, shit happen, right? Covalent modification is a much more permanent and effective form of modification rather than allosteric. Why is that? Because allosteric is based off of concentration. That's pretty simple. We got that. That's not bad. And genetic regulation is the most potent way of regulation. Yeah, that's pretty simple, right? If you have 10 copies versus 1 copy, well, 1 copy is going to work a whole lot less as well as 10 copies. Easy peasy. Signal transduction of insulin and glucagon in the regulation of hepatic glycolysis. PFK-2 and FBP-ACE-2, regulation of F26 bisphosphate concentration, and thereby the allosteric regulation of PFK1 and FBP-ACE-1. Covalent modification of hepatic pyruvate kinase by active pKa strongly inhibits PEP conversion of pyruvate during fasting in the liver. Why would we inhibit PEP to go to pyruvate?
And the fasting state is more glucose.
Yeah, more glucose. What's important?
Irreversible.
Yeah, why?
Because you're adding a phosphate to it.
Yeah, so you're breaking energy down. It kind of sucks to
You're
do.
removing a phosphate.
Or yeah, you're removing a phosphate, so you're getting energy, but yeah, you get the point. PFK2 and FBPase-2, the bifunctional enzyme, we're pretty solid on that. Okay, it's a super duper important regulator because it changes the concentration so that the allosteric regulation is impacted through the use of covalent modification by hormones coming throughout the blood. That's kind of the important part about that. I think you guys get that. Hepatic glycolysis, yeah, whatever. Okay, reduction of DHAP to glycerol phosphate regenerates NAD plus to keep glycolysis running and allows the energy of reducing hydrogens in the cytosolic NADH to support the ETC and ATP synthesis. Let's start with that half first. DHAP to G3P or glycerol phosphate. Yeah, that's not bad. It's a reduction, so it loses a hydrogen, right? Regenerates available NAD plus. Yeah, cool, easy peasy. To keep glycolysis running. What is another way we regenerate NAD plus? Lactic fermentation, 100% correct, and allows the energy from the reducing hydrogens in cytosolic NADH to support the ETC and ATP synthase. That's easy, you guys all understand that. Compare NAD plus dependent GPD1, which is a reversible reaction of the cytosol, against FAD dependent GPD2, an irreversible reaction in the intermitochondrial matrix. Note at the end, the reduction of pyruvate to lactate is also a means to regenerate NAD plus to keep glycolysis running. Okay, so we already said that one. First one, GPD1, which requires NAD plus, and FAD dependent GPD2. Should we go through that? Would that be a good place to start? GPD2. This is DHAP to glycerol phosphates. Let me pull up my notes for this.
And lose it by the
Okay, this works. Okay, so GPDH1, or glycerol phosphate dehydrogenase 1, takes DHAP and turns it into glycerol phosphates. And that's what? NADH to NAD plus. And the whole important part of this is that this can eventually go to tags. It's the glycerol backbone, as I'm sure you guys know. Oops. Well, over here, we have DHAP, and it works to make glycerol phosphate as well. But it uses the FAD. But is it FADH that goes into the yeah,
Yeah.
right?
So is it the conversion? So does it still use NADH for glycerol phosphate and then it uses FADH later on in the ETC, or?
So FADH, in what you guys showed me, it just goes directly into the electron transport chain, which is part of the inner mitochondrial matrix or membrane.
Because I'm just looking at,
Yeah.
so, because it would still use the hydrogens for glycerol phosphate.
Oh, and then you're saying the secondary portion of that reaction is the
FAD to
plus.
FADH.
Oh, I see. That sounds good. I like that more.
Well, because I didn't know if it was
No,
two
what you're saying
separate.
makes a whole lot of sense. Is this just FAD? Yeah, it's FADH. Okay. Because you have the glycerol phosphate, and we're saying that the glycerol phosphate crosses.
And then glycerol phosphate going back into DHAP uses, yeah, FADH.
Cool.
Because that's what I was just trying to
Yeah.
understand. But no, this makes.
Yeah. Just some kind of important to know is that you can also make triacylglycerides, Because this makes your glycerol backbone
Mhm.
of your triacylglycerides and stuff. And then you're saying glycerol phosphate. Yeah, okay, that makes a whole lot more sense. Sweet. Awesome. Cool. What's next? Where's my file? Here it is.
That's it. Perfect.
Yeah, I mean, let's read it. Reduction of DHAP to glycerol phosphate regenerates an NAD plus to keep glycolysis running and allows the energies from reducing hydrogen to the cytosolic NADH to support the electron transport chain and ATP synthase. Compare NAD plus dependent GPD1 reversible against FAD dependent GPD2 irreversible. Okay, so you're saying, like, what's important about this is that you're freeing this up to return to glycolysis while also making this glycerol phosphate, which would then turn back into DHAP, but allows one use of an FADH2, which is 1.5 ATP, right? I think it's 1.5 for his class. Because,
I don't think he's ever asked us about like what amount of
cool,
ATP.
then don't worry about it.
And I hope he doesn't.
So
ask
then most of the time, if you're considering this to be in a point where you're ripping through glycolysis and you need extra NAD plus to keep glycolysis going, and you would stop anything going into triacylglycerides. Did anyone tell Will where we are?
Yeah.
Also note that the reduction of pyruvate to lactate also is a means to regenerate NADH, or NAD+. Okay, yeah, whatever. Okay, PDH complex enzymes, E1, 2, and 3, plus the 5 required cofactors, TPP, alpha-lipoic acid, CoA, SH, FAD, and NAD+. Regulation of the PDH complex by activation of the specific PDH kinase by increased concentration of acetyl-CoA, ATP, or NADH inside the mitochondrion. Do not forget the opposing kinetic activity of the PDH phosphatase.
And I sent you a doc that has that written out too.
Oh, all right. Do you guys understand what the PDH complex is? Like what's happening?
It's 3 enzymes with 5 cofactors involved.
Yeah, and
Pyruvate turning into acetyl-CoA and NADH.
yeah, and you recognize that acetyl-CoA will then inhibit the production of itself.
To turn into octocetate.
Cool.
Quick question about that.
Yeah.
Why is it called PDH if there's no dehydrogenase involved? If it's all decarboxylase and then some other things. It's just that's how it is.
Well, because
They just name shit for shit, dude.
well, I do think
Okay.
it's because the NADH that comes off of it. That's the dehydrogenase part of it.
I think so, but the first enzyme is decarboxylase.
Pyruvate decarboxylase.
Yeah.
Well, no,
Or
that's the
pyruvate
second one.
carboxylase?
That's
One
the
of the two.
pyruvate carboxylase will be the second one because that goes into oxaloacetate.
And is that the one that gets inhibited?
Yes. Well, no, it'll be here.
The first one gets inhibited, right?
The first one gets inhibited
So,
by acetyl-CoA, which
PDH.
is decarboxylase.
Acetyl-CoA, which
Yeah,
goes
yeah.
back to inhibited. Then it goes to pyruvate carboxylase.
Where pyruvate decarboxylase is the first enzyme. I'm like 99% sure.
Sure, because pyruvate carboxylase.
I have pyruvate decarboxylase as the first enzyme,
Yeah.
which gets inhibited by acetyl-CoA.
Yeah, because that's what's in the textbook as well.
Yeah, which requires TPP or thiamine
And that's
pyrophosphates,
the only one that's
which is
required.
just a thiamine with 2 phosphates on it.
Yeah.
Because this one
Dihydrolipol.
is already pre-inhibited.
Because if we go to that page that we had, this one? Nope. Yeah, the one. This is pH and TCA. If you go up, this is enzyme 1.
And then I
enzyme 2,
have-
and then enzyme
Dihydrolipol acetylase, and then finally dihydrolipol
Oh, so I was
dehydrogenase.
thinking about this up here when it was down here.
3. Ah,
Okay, that makes sense. That's
pH phosphatase.
A month and a half. Never have to look at this again.
You'll have to see it again.
I know, but in my mind, I'll never see it again.
So, pyruvate dehydrogenase phosphatase is what reactivates the PDH complex.
Sorry, one more time.
Pyruvate dehydrogenase phosphatase is what reactivates the PDH complex. It removes the phosphate group.
And that's stimulated by calcium.
Oh yeah, actually I did know that.
Yeah, you did.
I think he had that in one of his notes.
Is one of the slides.
Yeah.
Yeah.
Because PDH kinase is what gets activated by acetyl-CoA and ATP and NADH to inactivate the PDH complex.
Yes. PDH phosphatase
So it's like, hey, we have a
activates
product.
it, and then
Let's-
PDH
let's slow
kinase
it down.
stops it down.
Yeah, so you have your PDH complex.
It's this thing here. Kinase versus the phosphatase, which acetyl-CoA stops it, and DH stops it, and ATP stops it.
Right. Yeah. Yeah. So this is kind of another, it's not the same as the bifunctional enzyme, but it's kind of similar. Where in the fact that you have allosteric regulation of enzymes that end up being covalent modifiers. So that's kind of an important thing to see here. You see how that's similar? You have allosteric modifiers that regulate these covalently modifying enzymes for a very large process. Just similar to the bifunctional enzyme that we talked about earlier.
Yeah.
Just an important thing to notice.
So how do we tie that in with insulin and glucagon?
Good question.
I would say this one. Well, what produces NADH, ATP, that pretty much be.
PDH cycle.
Well, like if you go all the way back up here, what stimulates glycolysis to go down further, which would be glucagon.
So the real importance of insulin and glucagon and the PP-1 and the pKa is to mostly regulate with the bifunctional enzyme and direct traffic to glycogen, right?
Yeah.
So it's one of those two, and that's kind of at the mouth of the river. So if you shut off the river, the rest of it's gonna trickle out. You know what I mean? So yeah, like technically you can even look at pyruvate kinase, where it's activated by F16BP and deactivated by ATP. But
I see.
you know what I mean?
It's not like a direct effect. It's like more like a secondary kind of thing.
Exactly.
That's exactly the kind of question you would ask.
Yep.
Yeah. Because I was like,
In a fed stable with
he had so many questions.
PDH complex.
He had so many questions that were like free, but that people overthink for like cyclic AMP. What is exactly? How does cyclic AMP get activated?
Yeah, through
And
the
then
denolate
it
cycle.
was like allosterically activated by changing the structural enzyme kind of thing.
Mhm.
Not like that would be the answer, but like things like that.
Yeah. I feel you. It kind of gets tricky. Tricky dicky. Okay. That leads us to what? 5? Yeah. Activation of pyruvate carboxylase, which is biotin dependent, by increased concentrations of acetyl-CoA inside the mitochondria. Why does this happen in the fed state in the liver? Okay. Pyruvate carboxylase makes oxaloacetate, right? And it is increased by concentrations of acetyl-CoA inside the mitochondrion. Okay, so we kind of talked about this already. Acetyl-CoA will, will inhibit the PDH complex through the use of PDH kinase. So if PDH kinase inhibits the PDH complex, your only real other option is pyruvate carboxylase, which is also stimulated by acetyl-CoA. So if you have stimulation and the other path you're closing off, then you're gonna really go down that one. Inside the mitochondria. Why does this happen in the fed state in the liver? The fuck? Fed state? What does that have to do with anything?
He said because in the fed state you're making more pyruvate because you're insulin, so you make more
Yeah,
pyruvate, so
that makes sense.
increased levels of acetyl-CoA, which inhibits itself.
Oh, yeah. Yeah, that makes sense.
Yeah.
When you're ripping through it, you're making a shit ton of
Exactly.
acetyl-CoA and
High levels of glucose, more
Yeah.
pyruvate. That's what he said, but
yeah, that makes good sense.
yeah.
Does that make sense to you guys?
Yeah.
Yeah. Okay. Next one. Citrate is synthesized and has two fates: either be oxidized to isocitrate through the TCA cycle or build up to elevated concentrations inside the mitochondrion and be transported out into the cytosol, where the liver will use the citrate during de novo fatty acid and cholesterol synthesis pathways. Okay, so let's talk about that real quick. Citrate is crazy. Citrate is a super duper late down the line product that actually comes way back and inhibits PFK-1. That's important. Don't forget that. So citrate can also just go into isocitrate. Isocitrate dehydrogenase is regulated by ATP and NADH. So when you have a high amount of energy already, you don't want to make more.
Slow it down.
Yeah, so you're going to inhibit isocitrate dehydrogenase. If you inhibit isocitrate dehydrogenase, you're increasing the concentration of isocitrate.
Which then even slows down production of acetyl-CoA.
Exactly.
So it's a double.
Exactly.
Sorry, I didn't clock that. I lost.
So here.
I lost.
So isocitrate dehydrogenase, it's inhibited by high energy yield. So if you have so much energy, you don't need more. Okay, take that same thing, move it over here into slowing down PDH. So now it's double inhibiting energy reduction.
Because you have a shit ton of citrate now, and that citrate is going to flow out of the mitochondria. It's going to go all the way back to PFK-1. I mean, yeah, PFK-1. So you're going to inhibit from the top, and now you're all the way down to.
It inhibits close up and form.
Mm-hmm. Now you're continuing to make fucking acetyl-CoA, right? And your acetyl-CoA is going to turn into malonyl-CoA or malonyl, and it's going to make palmitate, right? Everyone remembers palmate?
Huh?
Polonic acid?
Or palmitate, yeah, it's the same thing.
Oh, like a 16 carbon fatty acid.
Yep. Made from repeats of NADPH and NAD+, right? No idea? Okay, cool.
We just got to
He probably talked about
citrate
it.
back to pyruvate, citrate to, was it?
Isocitrate?
Malonyl-CoA?
Yeah, malonyl-CoA.
And then that's put into each step for, I guess, building fatty acids. But
Yeah.
we
Fatty
only,
acid synthase?
like, we stopped there, and then it was
Okay.
glycerol phosphate going into tags.
Oh, okay. So let's quickly chat about it just to give you guys some answers about that. Your acetyl-CoA is going to go into fatty acid synthase. You need your first one to be an acetyl-CoA. After that, you need a shit ton of malonyl-CoAs. How do you get that? Well, through acetyl-CoA. How am I getting acetyl-CoA? Through pyruvate. How am I getting pyruvate to acetyl-CoA? Through the PDH complex. Now, if I'm ripping through the use of acetyl-CoA to malonyl and putting it into the fatty acid synthase, then it's lowering the concentrations of acetyl-CoA, which is allowing you to make more of it, even though we just talked about how high concentrations of it is going to inhibit itself. So in this case, we're making it and then we're instantly changing it to something else to falsely kind of lower the concentration of it, allowing for more of it to be made. Cool. Now we're making a shit ton of it, and what we're doing is we're taking the malonyl and we're adding it to the synthase, fatty acid synthase. To kind of break down what that looks like.
So it's kind of like it's like phosphodiesterase activating cyclic AMP back into AMP.
Gotcha. So does that make sense? Higher energy, like fascistic molecules activate. So here is an NADH concentration increase in this regulates pH complex. So can I say high energy EO molecules activate pH kinase which limits pH complex?
Yeah, that's right.
Yeah.
Then how are you getting more of that?
But more energy.
Yeah, so through your TCA cycle. So if I'm going through my TCA cycle, then I have a shit ton more of ATP and high amounts of NADH, which are kind of like the precursor to that high energy. And because of that, I'm going to inhibit isocitrate dehydrogenase. Cool.
And
We don't have to make- we don't need more.
Mm-hmm.
Which is PFA, so it's a double inhibition. Got you. Clocked it down.
That's what I was saying.
And then if we do that,
Makes sense.
we want to make fats now. And how are we going to make fats? Well, we're going to make it through citrate or fatty acid synthase. How are we going to start fatty acid synthase? Well, we need to prime it. How are we going to prime it? We're going to use an acetyl-CoA.
Is this why it gets broken up into oxaloacetate and acetyl-CoA when it gets out?
Yeah.
Gotcha.
So now you can use it again, right? So this is my citrate synthase, and I'm adding an acetyl-CoA, right? And then now it's primed. Now it can start working. So then I'm going to add on my malonyl, ALONYL, right? And what ends up happening during this chain extension is I add it on up here, and then they swap down, and then I add a new one up here, and then put it on, and then back up. Like it's
Keeps on using acetyl-CoA and malonyl-CoA.
only malonyl. The first one that you need is acetyl-CoA to prime it.
Oh, so you just keep on adding a bunch of malonyl
Mhm,
which
mhm.
you have to break acetyl-CoA to make malonyl-CoA.
You just have to add a phosphate, I think. It's not a big difference.
Oh, okay.
Yeah.
It's ATP
And
breaks up
carbon
to
dioxide.
Yep.
Gotcha.
So you need a carbon dioxide and you need ATP, which really just gives a phosphate. So it's structurally different enough to where you're lowering the concentration of acetyl-CoA, which makes the formation of it faster, and you're storing it in this usable form that you're going to use for this. So you need to extend the chain out further, but every time you extend the chain out further, you need an ADPH, and you need a lot of it because you need to donate the hydrogens to saturate the fat. So you have palmitic acid, which is a 16 carbon saturated fatty acid, right? So you need a shit ton of these hydrogens, which you're getting from the NADPH. Where do I make NADPH? Not the TCA.
No, not TCA. That would be
HMP, hexose monosphosphate. Yeah,
The first step.
yeah,
Can we do that after?
yeah.
Because that's Pellagra.
Yeah, Pellagra. Okay, now what is the other option is cholesterol, right? Right? So how do I make cholesterol? Cholesterol, I add a shit ton of acetyl-CoAs. Like basically I bind two together, so I get like a 4 carbon chain, and then I add 2 of those 4 carbons together, and I add 2 of those 8 carbons together, right? It's kind of like this,
Stacking.
right? Going nuts with it. What's interesting is that it uses HMG-CoA synthase.
Yeah, that right there.
Uses HMG-CoA Okay, synthase. Which is potently inhibited by PK. Okay. How do I activate PKA?
Kinase A, so that'll be
Cyclic AMP,
Cyclic AMP, which is made from
pentanoate cyclase.
glucagon. Yeah.
Technically, I'm right.
Yeah.
But now what we're looking.
So if I have glucagon, it's because I have high or low blood sugar? Low blood sugar. So if I have low blood sugar, then I don't want to be making fats, right?
So this would be low
Mhm.
blood. Yeah. So this would be in.
Biogenesis, beta oxidation.
This would be the fastest thing.
Yeah. So good job. That's not bad, right?
Yeah.
To be honest, it gets real bad when you go through all of the cholesterol synthesis, de novo synthesis of cholesterol,
Is it
because you
is
have,
it all that right there?
yeah, it's all that actually.
He would ask us that on this midterm.
I remember he said something about like IPP, which is isopentenyl pyrophosphate.
Yeah, there you go.
I think it was because that required ATP and let go of a carbon dioxide. Or you use IPP in a ton of other reactions too.
I don't think I've ever
Yes,
read
you
that
do.
before.
IPP goes in I think it's the next reaction
And the one after that too.
is dimethylallyl.
Dimethylallyl pyrophosphate, which uses another IPP. IPP is basically like this carbon donator in a way. So think of it as a large strand.
Kind of like salmon salt, or donate a methyl group.
Yeah, except much larger scale. If you wanted to think about it, you know those stacker games, like Stack the Tower,
Yep.
Yeah.
you have the arcades? You're basically, like you have this layer, and then you're taking another layer and you're stacking on top of it. And you're taking another layer and you're stacking on top of it. Another layer and you're stacking on top of it, because you want to get that structure of cholesterol, which is what the, whoops, something like this, right? Right, something of the sort, and then you have like a pentose.
Sure.
Because it's like A, B, C, and D. You know what I'm talking about?
Mm-hmm.
Those are like steroids, but the steroids are from cholesterol.
Today in class, you did say that there wouldn't be any questions on triglycerides or fats or anything like that.
Oh, dude, kick ass.
So, I'm not sure.
Okay, so then know the basics of it.
No base got you.
Know like, hey, what the fuck is going on?
Like, for example, that one makes sense.
Like this?
Acetyl-CoA one, like, because that's how you make oxaloacetate, because you're using acetyl-CoA. So,
Mhm.
how to make more acetyl-CoA until it inhibits itself, and then oxaloacetate takes over.
Yeah.
Gotcha.
And also like, well, now if I have a shit ton of acetyl-CoA,
Yeah.
then what? Well, I'm gonna stimulate PDH kinase. My PDH kinase is gonna inhibit the PDH complex.
Yeah.
We're going to have philosophy.
And it's also going to inhibit, lots of acetyl-CoA.
And then that can
Citrate.
create more citrate.
And if I have a shit ton more citrate, then citrate's gonna go up and it's gonna inhibit PFK-1.
PFK-1, and then
Which
you can
stops
even go back into
all of it.
oxaloacetate and acetyl-CoA, and that will start fatty acid synthesis.
Okay, cool. So let's read the question again. Citrate is synthesized and has two fates. Either be oxidized through the TCA cycle, perfect, we know that one. Isocitrate to alpha ketoglutarate to, succinate, succinyl-CoA, and then fumarate, malate, oxaloacetate, back to, easy. Or build up to be elevated concentrations inside the mitochondria and be transported out into the cytosol. Okay, then what? First thing we're gonna do, we're probably gonna inhibit PFK-1.
Yeah.
Where we're gonna use it in de novo fatty acid and cholesterol synthesis pathways. Okay, we just quickly went over de novo synthesis of fatty acids and de novo synthesis of cholesterol. Cool. Okay, next. TCA cycle regulation, specifically ADP and ATP effects on the activity of isocitrate dehydrogenase. How the inhibition of isocitrate dehydrogenase causes citrate to accumulate and thus be transported out of the mitochondria. So real quick on that, citrate and isocitrate are what of each other?
The same
Isomers. What's an isomer?
That's just horrifying.
Comes in, directly right answer right away. This guy's just a genius. What?
They're just rearranged.
They're just rearranged, right? So are isomers like at the same concentration of each other?
Yes.
Yeah. So they're always going to be at the same concentration of each other.
That's the equivalent. Like,
Yeah.
That's the
So like if you have 100 copies of citrate, 50 of them are going to turn into isocitrate, the other 50 are going to remain citrate, give or take, right? So that's really important because if I'm using isocitrate, my citrate levels are going to rise. So what are they going to do? Well, they're going to turn into isocitrate to stable it back out, right? That stabilization is because I'm having more citrate come in through oxaloacetate and acetyl-CoA. Easy, not bad. Cool. So what's important? Well, now I'm continuing to lower the levels, and the levels are continuing to rise on the other side, so they're just equalizing. But now, instead of removing isocitrate, I'm keeping it the same. So I'm still having this big buildup of citrate, and my isocitrate is going to rise as well. Because of that, it's like I'm going to hit max capacity, and they're going to exit the fucking mitochondria. Why would it exit? What's kind of a big deal?
What's going to exit citrate?
Yeah, citrate's going to exit the mitochondria.
So to stop the production of.
Yeah, so it does stop the production, but why can't I just leave it in the mitochondria? What's the big deal? Yeah, it swells up. Why?
Just on finite room.
Is it a base or is it an acid?
Acid.
It's an acid, right? These are all acids, right? What do acids do with water? Hold on to them or push them away? It holds on to it, right? It brings more water in. That's why whenever people have a shit ton of glucose, right? Water is going to follow the glucose, and these people that have a shit ton of glucose are going to be swollen or edemic because they have a shit ton of water in their system. So if my citrate stays in the mitochondria, it's going to pull in more and more and more and more water. And if I have more and more water inside my mitochondria, what it's going to do is it's going to blow up. How bad is that? Well, that's what's terminal. It's over from there. You're not doing anything to fix that. It's game over for the cell, right? So it's going to go into the cytosol where it has it doesn't matter how much citrate you have in the cytosol because your cytosol is like the ocean. If you have a shit ton of citrate in the ocean, what's it going to do? Nothing. It don't matter, right? But if I have a shit ton of citrate in like the kiddie pool, well, that shit's going to overflow real fast. You see the difference in those two? Cool. How the inhibition of isocitrate dehydrogenase causes citrate to accumulate, and thus be transported out of the mitochondria. Done. Hypoxia and the inhibition of the ETC, complex 4, NADH concentration increases, and this regulates PDH complex, alpha ketoglutarate dehydrogenase complex, and isocitrate dehydrogenase. Okay, why is oxygen important in the ETC?
Changes into water, accepts that final electron receptor.
Yeah, it's the final electron acceptor. That's kind of the really super important part about that. So that's a really big deal. If I don't have this final electron acceptor, can I use anything else in its place? No, I can't. Cool aside, the stuff at the volcanic pits in the middle of the ocean, they use sulfur.
Don't even talk about sulfur right now.
Element of life.
You either.
It's like saying Bloody Mary 3 times. Gail's gonna show up.
No.
Get out of here. Okay. Cool. So the final electron acceptor is oxygen. If you don't have oxygen, you're out of luck. Let's say I don't have oxygen. What am I gonna do to make energy?
Sorry, one more time?
Okay, I'm out of oxygen,
Yeah.
or mostly out of oxygen.
Anaerobic glycolysis. Lactate.
Glycolysis. Yeah, I'm just gonna rip through glycolysis. And then I'm gonna make what?
NAD plus.
Yeah, and FADH.
Okay, let me rephrase. I'm out of oxygen, okay, or I don't have enough levels to support the TCA cycle, or the ETC, which in turn means the TCA cycle, right? I can't go through the TCA cycle, so I'm not changing pyruvate into acetyl-CoA or citrate or oxaloacetate. It's going to lactate, right? That's really important because we need the NAD plus to be regenerated, which you guys know. That lactate, where do I put it? Where do I put the lactate once I make it?
Oh, I know this one. Buckets of process. Yeah.
My veins, bro. Blood. I put it in my blood because that goes to my liver, and the liver takes care of it. Right? So then I still have high concentrations of glucose in my muscles for them to rip through it, create more lactate, and put that back into the blood. If I have a high amount of lactate in my blood in a certain local area, what is going to happen to the local pH?
Decrease because it's an acid.
Nice, it's going to decrease because it's an acid. If I decrease the local pH of the blood in that area, what is going to happen to the hemoglobin?
Oh, super taut. Doesn't release oxygen. Oh, wait.
The pH decreases.
If the pH decreases, then I'm protonating the histidine residues. If I'm protonating the histidine residues at a much higher rate, then I'm going to have much more stabilized taut structure, which is the release of oxygen. So I'm going to release even more oxygen to that oxygen-hungry tissue already.
Does that kind of fix itself though?
Will it kind of fix itself? Yeah,
Yeah.
that's kind of the point.
Oh, it's trying to fix itself. Got
Yeah.
you. I see.
Because you don't have enough oxygen,
Yeah,
right?
yeah, yeah.
So I have no oxygen, so I'm making more lactic acid. The lactic acid goes into the blood pH is lowered. The pH being lowered hits the pKa of the histidine gets protonated. Protonated histidine changes the form to the taut form. The taut form favors the release of the oxygen. I have 4 oxygens, right, on the heme. So I need to release all 4. And if that's not enough oxygen, then we're kind of shit out of luck.
I have a question about that though.
Yeah.
If you're in a state of hypoxia, how are you going to get access to more oxygen to refill those blood cells?
Good question. You remember the oxygen hemoglobin dissociation curve?
Yeah.
Do you remember how much percent association your venous blood is?
37%, 27%, whatever it was.
Kind of.
I swear it's like millimeters per mercury. Like, I don't remember.
So this is the percent association, and this is the partial pressure O2,
Oh yeah.
right? So it looks like this.
30. It's like 20 to 30.
Yeah, but don't even worry about this. Worry about the association. So in our venous blood, at the most venous that we get, it's probably like around here, which is, let's say, 80%.
Yeah.
So if we get hypoxic,
It goes
the
down.
partial pressure of O2 is going to decrease, which means we're going to go to like, let's say here, let's say that's 50%. So we're still able to rip more oxygen off of the hemoglobin.
That makes sense. So instead of just releasing 1, for example, it released 2 or 3. Got you.
Yeah,
Nice.
isn't that kind of neat?
So he's gonna ask you the question of partial pressure.
Yep. Because
He did mention that, saying, yeah, I can test you from Biochem 1 stuff.
Yeah, so that's Biochem 1 stuff.
Yeah, no, I understand that.
Yeah, but do you see how they all fit together
It
now?
interchanges.
What number is this? This is number 7, hypoxia. Number 7, hypoxia.
Yeah, and also what I have.
Quick summary of what I can
have
say to
this,
that.
which is
Hypoxia.
So
The whole
hypoxia
view.
is the lack of oxygen,
Lack of oxygen.
Because
which means my
during-
final electron carrier in the ETC
I'll
does not
listen
exist.
to you after this. Because I wrote this from his.
Electron carrier.
So the glycerol phosphate shuttle is
If it doesn't exist, then
Combining that
what's
with-
going to rise?
this right here.
My high energy carriers, right? My
So we
NADH
get
and my FADH2.
complex
So I'm gonna have an
protein.
increase in the concentration of NADH
Or
and
this is
FADH2,
like intermediate protein.
Yeah.
which is in turn going to
So
inhibit
C complex 1, 2, 3.
alpha ketoglutarate dehydrogenase
This protein
complex
is an intermediate.
and isocitrate dehydrogenase.
In a membrane space.
If I inhibit those two, I'm gaining more
alpha
citrate,
glycerol phosphate.
right? And then from there,
Go
you know where that goes,
to
right?
Protein.
High levels of
Give
citrate
off
Yeah.
2 hydrogens
inhibits
that it got from here.
Bump glycolysis and stuff, goes to
Yeah.
lactic
Which will
acid,
then be
lactic acid,
used
then causes,
To funnel
phosphate,
electrons
phosphate,
and hydrogens into that
Exactly.
membrane space.
Nice.
Thank you.
You're welcome.
So I had to like reorganize some of his notes, but one of his slides for ETC was this, word for word.
Staying out of your picture.
Thank you, sir.
I don't
What's
wanna
going
be
on your
featured.
YouTube channel?
It is going on the YouTube.
Are you gonna take my voice out of it, or
No.
are you gonna continue? Are you gonna mute me or no?
Oh, it's rolling, baby. Ain't no stopping the train.
I should probably stop cursing.
Wait, so it inhibits that cycle, which in turn stops citrate
Production.
production? So then lactic acid takes over.
And if you remember, here, let's do a quick little. So if my ETC has no oxygen, right, it's going to inhibit the TCA through the use of isocitrate dehydrogenase and the alpha ketoglutarate dehydrogenase complex, right? Which is then going to cause a buildup in the concentration of citrates.
And then backs up over the top.
If I have a high concentration of citrate, it's going to leave and turn into what? Malate or acetyl-CoA and oxaloacetate, right? My oxaloacetate can go back into pyruvate, right? Because it goes malate, fumarate, or fumarate, malate, and back to pyruvate. Yeah.
Mm-hmm.
My acetyl-CoA is going to inhibit PDH complex, which is going to inhibit the creation of itself, but stimulate creation of more oxaloacetate, right? If I'm stimulating more oxaloacetate, then the more oxaloacetate can go back to pyruvate, basically. And if I have more pyruvate, then it can go back and forth with lactate, making a higher concentration of NAD plus, because over here, I have a high concentration of NADH, which is inhibiting the TCA. Picking up what I'm putting down?
Yeah, we're good.
I completely if I'm gonna be completely honest with you.
That's okay.
I'm tired.
Let's go slow. Okay,
No more influences.
my electron transport chain, what does it need?
Oxygen
It needs oxygen. Why does it need oxygen?
to make it ATP, or the final electron carrier.
It's the final electron acceptor. That's what's important about it. It doesn't need it to make ATP, it needs it to get rid of the trash. That's kind of the whole idea of it. Okay, so So if it doesn't have oxygen, it can't run. If it can't run, its substrates are going to build up.
NADH and FADH.
So increase NADH, FADH to, does that check out so far?
Yeah.
It's okay if it doesn't, maybe.
It's alright, I'm just tired.
If I have an increase in these, then I want to stop their production.
Right.
How do I stop their production? Well, the TCA is what produces all of them. So I need to stop the TCA. Specifically, I do that through stopping alpha-ketoglutarate dehydrogenase complex and isocitrate dehydrogenase. Now, if I inhibit both of those, then I'm going to have a buildup of citrate.
What exactly does alpha ketoglutarate dehydrogenase do again? Oh my god, never mind. That turns that into isocitrate, which then turns it back into citrate.
So it goes isocitrate to alpha-ketoglutarate?
Yeah.
Yeah, you got it.
That's not bad. Sorry.
Oh, good, Ben. Now, if I have a high amount of citrate, citrate is both of these, but which one can we really use more? This one.
So it turns back into PEP.
So we get both of these. So this is going to, at high concentrations, inhibits itself. So it's going to stop the making of itself and stimulate the making of its cool buddy. Yeah? OAA?
Can you draw an increase in acetyl-CoA?
Yes.
Thank you.
Where do you want it?
Beside the acetyl-CoA, like where you put the inhibit, like you put increased concentration of acetyl. Thank you. Thank you.
Yep. Cool. Stop. I want no more of this. Make more of this. Yeah? More of this means what? More of this means more of this. More of this means more of this. More of this means more of this, which allows more of glycolysis to happen. Glycolysis is where I'm getting my energy in anaerobic state.
But then how does that fix hypoxia?
It doesn't.
It just makes sure the system keeps going.
It gives you energy. It's like turning on the generator when the power's out. It's only going to last for as long as you got the fuel.
Ah.
It's not going to fix it, but
It
it
just
keeps you
takes
alive.
care of itself until the hypoxia stops.
Yeah. So, how does the hypoxia stop? You stop working out, right? Your muscles literally turn off, or
Stop
you
being choked,
die.
swimming underwater.
Yeah, whatever it is, you just die.
Ah.
Easy way out.
Take
Just
your
die.
pictures.
Yeah. I'll make sure it gets on the vidya.
So when there's no oxygen, you have a buildup of what, FADH and NADH?
Yes. Why?
Because you're not able to transfer that hydrogen to the oxygen, so if there's no acceptor, the hydrogen's just built.
So you're getting slightly confused.
So part to self is, remember, NADH goes through complex 1, which then gives out its hydrogens. So if it can't go through complex 1, which gives its electron, which gives it the power to go through, it would just accumulate.
So technically you could continue making the hydrogens go through. You could totally continue to make the NADH just into NAD+, and you could do the exact same thing with the FADH too. You could keep using them as their intended purpose. But what's gonna happen? You're gonna have electrons that are gonna build up. And what are these electrons gonna do? They're basically like your free radicals, which is, for lack of a better term, like your cell terrorists. And that is very bad. So you don't want that to happen ever. So what are you gonna do? You're just gonna not do it, right?
So just buildup of, let's say, the hydrogen ions, and it just accumulates.
So let's draw it. So this can be 1, 2, 3, and 4. And And this is ATP synthase, right? So over here is where you get your oxygen. And your electron starts off over here. Yeah, how does it get started? From this. So they're just using the energy of these reactions to kind of power or propel the electron forward. The electron is a third party in this case. Does that make more sense now?
Yeah.
Cool. Next? Nope. Yeah.
Back to number 7, it says specifically ADP and ATP effects on the activity of isocitrate dehydrogenase. So I get that if you increase ADP levels, because isocitrate dehydrogenase, when it's activated, lets out ADP. So that accumulates citrate. Let's just say under normal conditions, what would citrate, what does that do?
What does citrate do under normal conditions?
Under normal conditions.
It just lives.
Oh, okay.
It's just in the
It would just be
mitochondria.
cholesterol and fatty acids. That's it.
Oh, are you talking about in terms of what it get used up in a separate case?
Or like here it just says, if you look at number 7,
Yeah.
it says isocitrate dehydrogenase, and then I said like more citrate goes turn off PFK1 and stops glycolysis. That's what citrate does, right? When
Yeah.
there's excess of it.
Yeah.
What exactly else does it do? Or does it just do that?
Well, it gets used. Like citrate itself gets used.
Yeah, but like
TCS
it says
ADP and pH on the
transport
active
out the
concentration.
mitochondria.
Yeah. So once it goes out of the mitochondria, it inhibits the PFK-1 and it goes back into OA and acetyl-CoA. So it gets broken.
Okay, and then
And then-
acetyl gets used into that.
Yeah, or you could just make citrate again if you wanted.
Okay.
Picking up what I'm putting down.
Yeah, because I understand under hypoxic conditions, that's what's happening. But
This is
under-
also under conditions where you're just inhibiting this. It's like take this completely out of those
or
situations.
just inhibition. Got you.
Like let's say you have high energy. Like I was working out. Am I working out anymore? No. What am I doing as a result? Well, now I have a shit ton of energy left over because I was prepping to keep going for another 6 miles. But Lord knows my fat ass ain't about to go another 6 miles because fuck that.
I see. And that citrate just chills it or breaks up, or it could just chill there in the cytosol, or just,
Yeah, and you're always making more ATP
yeah,
because you always need it.
yeah.
So you're just gonna you're gonna stop making a shit ton of it. You're just gonna slow it to like the trickle that you need,
Yeah.
and then you'll use the supply for the TCA, or you can just change it back into acetyl-CoA and acyl acetate, which can go their own way and do other things.
Gotcha. So OA can only go to TCA or pyruvate. There's nothing else it can do. Or like
In this case, yes.
in this case, yes.
While acetyl-CoA is more of the
Important one. Because the
opportunistic, because it can go to malonyl and cholesterol and de novo fatty acid.
Gotcha. So let's be trying to box inhibition.
So let's read that from the top. TCA cycle regulation, specifically ADP and ATP effects on the activity of isocitrate dehydrogenase. ADP will stimulate it, ATP will inhibit it. Right? That's simple. How the inhibition of IDH causes citrate to accumulate because they're isomers and thus be transported out of the mitochondrion. Why? Lake versus, or little tiny kid pool versus ocean. Hypoxia and the inhibition of the ETC because complex 4, because it doesn't have the electron acceptor. NADH concentration is going to increase. As a result, that's going to inhibit PDH complex, alpha ketoglutarate dehydrogenase complex, and isocitrate dehydrogenase.
So high energy things activate that to stop that.
High energy stuff inhibit the creation of more energy. Either more energy intermediates, high energy intermediates, or energy itself. Q. Know the TCA cycle enzymes: citrate synthase, acheontase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase complex, succinyl-CoA synthetase, succinate dehydrogenase fumarate, and malate dehydrogenase, and the reactions they catalyze of the TCA cycle. Appreciate that oxaloacetate is the beginning and the end of the TCA cycle because it returns to the beginning after each turn of the cycle to form the next citrate from whatever macronutrient fuel provides acetyl-CoA. I mean, that's not bad, right? You guys should kind of know that from one.
Mm-hmm. Well, so like fumarate into malate, is that just a simple?
Yeah.
Right, because I'm just trying to find because all of them have either NAD plus and NADH, or
Yep.
there's.
So fumarate to malate with fumarase, it's just done. It happens.
Like there's no.
It's just like a
nothing comes
rearrangement.
off, or you could say it's like an isomerase almost, or.
I wouldn't say isomerase because it's not reversible. It's like removing a bond and putting it in a different area,
Okay, it's like
which is similar to isomerase, except this bond is more permanent.
Okay. Gotcha.
Does that check out?
And similar to oxaloacetate to citrate.
Yeah.
Okay.
Well, sorry, no. Why? Because oxaloacetate needs acetyl-CoA.
Okay. That's what's missing.
How does NADH concentration regulate PDH? Shouldn't it be NAD? Oh no, because NAD plus is what you need to continue it on. NADH would stop it.
You have to think
Backwards.
further.
Yeah.
Further steps. So NADH at high concentrations inhibits the creation of more NADH. Right? Because if I have a lot of something, I don't want any more.
Gotcha.
So what is that going to do? It's going to make more citrate. My citrate's going to go back and it's going to do all that fun stuff.
Makes a lot more sense now.
Okay. Oxaloacetate needs acetyl-CoA to become citrate. That's the important one. And most of them have some sort of extra thing happening, right? NADHs or FADHs or GTP.
GTP would be succinyl-CoA desuccinate.
Yep.
Yep.
And you're regenerating a CoA species, which you can put back on a pyruvate and make acetyl-CoA.
I hate how you connected those things.
Yeah. Okay. 9. Therefore, appreciate that carbohydrates, which provide pyruvate, are not the only metabolic fuels that will be oxidized in the mitochondria. Ketone bodies, fatty acids, alpha amino acids from proteins, once their amine nitrogens have been removed, can be oxidized as fuels in different cells of the organs and tissues. That's just like a statement. That's just like a statement, right? You guys all know that ketone bodies and all that other
Amino
shit
acids,
goes in
rate
there.
oxidation.
Yeah, that's not bad.
Gluconeogenesis, that's
Yep.
What happens in
Number 6 and 7 were the ones I was having a bit of a hard time understanding. But that makes a lot more sense now.
Okay. 10. Electron transport chain and oxidative phosphorylation. Complex 1. NADH dehydrogenase. The site of greatest translocation of H plus ions in the intermembrane space. Complex 2. Succinate dehydrogenase is FAD dependent. Both 1 and 2 provide reducing electrons and hydrogen to ubiquitone, UQ or CoQ10, to continue transport to complex 3. 3 contains redox cytochromes, heme proteins with different ferrous ions. Cytochrome C is bound to the outer leaf of the inner mitochondrial membrane, and when reduced, will diffuse across the surface to complex 4. Complex 4 has copper, Cu. What is Cu?
Yeah.
Yeah.
Copper.
Redox couples that transfer the electron from cytochrome C to an iron-based protein inside. Complex 4. Inside complex 4. What the fuck? Complex 4 is where the reduction of oxygen, O2 to H2O, occurs.
Put that shit down.
Do you guys want to go through that? It's just a lot of words to say.
Yeah.
Yeah, it's a
And
statement.
then
It's just talk about the
I send
electrons.
it to you if you want to look at it.
Cool.
But I'll send it to you as well.
11. H plus ions translocated out of the inner membrane space represent a high chemical potential. The energy of chemical potential is released as the H plus ions flow through ATP synthase protein structure of complex 5. Mechanical deformation of the protein structure of complex 5 occurs when the H plus ions flow through the F0 protein, causing F0 to rotate. Structure of F1 becomes strained as F0 rotates. Mechanical energy is released when deformed. Strained structure relaxes back to initial unstrained configuration and structure. The energy released by F1 is used to produce ATP from ADP, inorganic phosphate. Antiporter integral to inner mitochondrial membrane actively transport ATP out of the mitochondrial matrix to the cytosol, where ATP is transported from the cytosol to the matrix. This activated anti-transporter consumes 25% of the ATP formed by complex 5.
I have a question about that.
Yeah.
So basically, as F0 rotates, F1 gets like crumpled, for example, it's like crumpled on paper.
Mm-hmm.
Then the energy that's created from the rotation fixes the crumpled paper back to normal, in a sense? Is that what it's trying to say?
Yes and no. Do you know how this works? Like what's actually going on?
I know the self-is version of it.
Okay. So think of it like a water wheel.
Yeah.
When you put the water through the wheel, it starts spinning. And we can say that spinning is what's going to twist a rubber band. Now we twist this rubber band huge, like super twisted. Now it has a ton of this energy stored. ADP is going to come in. It's going to get the phosphate added, become ATP. That like untwists it.
That's what unstrains it, is the binding of ATP and ADP.
The making of ATP.
Sorry,
Yeah. So the using of that stored energy, if you will. Cool. And then once you make that, it instantly leaves, right? Because you need to go use it somewhere else. Cool?
So you could say it's like from Jackass, the big hand. The hydrogen just winds that shit up.
Exactly.
And then it just, bam.
Uh-huh. Okay. Any other questions, or are you guys feeling great?
What vitamins biotin?
I don't know.
E6.
I thought
B6?
that was
I
Niacin?
don't have it written.
PLP.
I do not have it written.
PLP?
I don't know. I would guess.
Oh, I'm pretty sure that's PLP.
I don't know.
Already, I'm